All right, hold on to your seats and pens as we do some more patterns of inheritance. Taking a look at some more details and some more complexities about genetic inheritance. Okay, so after learning about monohybrid and dihybrid crosses that Mendel did with his pea plants, we can actually use that phenomenon in order to determine the unknown genotype of a certain organism. So say we're riding down the road and we see a pea plant. Ooh, there's a pea plant. And it's got purple flowers. That's awesome. But remember, purple is dominant, so we wouldn't know if it was actually purebred purple, big P, or if it was heterozygous purple, big P, little p, right? So what we can do, grab that pea plant, we can take them home, and we can do what is called a test cross. Okay, we're going to cross an unknown genotype with a known genotype. Okay, so we usually cross a homozygous recessive during a test cross. So what we're going to do is we're going to cross this unknown purple pea plant that we found on the side of the road with a white pea plant, because we know white has to be homozygous recessive, little p, little p. Okay, so there's two possibilities that we can get for the F1 generation. We could get either, so if we see that all of the offspring from these two are all purple, right, then we know that this has to be big P, right? Because we have to have the dominant P's for all situations in our Punnett square to be all purple. Okay, so it's got to be big P. However, if the offspring come out to be purple, if we get some purple and some white ones, then we wouldn't say this would be big P, it would be big P, little p. Okay, because we're going to have little p from both parents in order to make a white pea plant in the end as well. So we can do this, what's called test cross, to figure out what the genotype is in the unknown plant. Okay, so here's just the slide so you can put down exactly what I said in the last slide into words. So if an unknown genotype is homozygous dominant, then all of the offspring would be heterozygous, big P, little p, and have purple flowers, just like we had said here. Okay. However, if the unknown genotype is heterozygous, big P, little p, then we're going to have offspring that are going to be big P, little p, and homozygous recessive with little p, and we are going to have some purple and some white. So that's one way we can use our genetics and our monohybrid crosses and dihybrid crosses to figure out unknown genotypes of particular phenotypes of organisms. So in genetics we have this rule of probability, which basically says when those gametes are forming, when those sperm cells and egg cells are forming, meiosis is happening, those alleles of genes, okay, the different alleles, the different forms of the genes, okay, whether it's dominant, recessive, they are going to segregate into the daughter cells basically kind of by random chance, okay, it obeys the rules of probability. So it's basically the same as tossing a coin. So in our rule of probability, each coin toss is an independent event. One coin toss doesn't affect the other ones or any previous ones. So the probability of getting heads is 1 out of 2, right? There's 2 sides to a coin, so there's 1 out of 2 possibilities of getting heads. When tossing 2 coins, right, the probability of getting heads is a product of both the tosses. So to get 2 heads in a row, the probability would be 1/2 times 1/2, which would be 1/4. So we call that the rule of multiplication. So this is basically how the segregation of alleles within meiosis occurs the same way as well. It's like flipping a coin. Which way is the dominant going to go, left or right? When we're looking at the meiosis, is it going to go into one cell or the other? what's the chances of the next one going? Well, again, it's one cell or the other. It's like 1 to 2. So we're going to see that we can use our genetic basis that Mendel described for us in our pea plants, our dominant versus recessive that we kind of know about, heterozygous, homozygous, and probability of segregation and independent assortment, to take a look and actually analyze kind of our own genetics that we see as human beings. So many human traits are going to be determined by simple dominant recessive inheritance. Okay, so these are some traits that are simply dominant or recessive. So some dominant traits: freckles. Yay, I have a dominant gene because I got freckles. Having a widow's peak, which is your hairline. Okay, so if you take a look at this woman right here, she's got a pointy hairline. That's what we call a widow's peak. Okay, and having free earlobes, okay, that can wiggle wobble, is again a dominant trait. Now, if you had no freckles, okay, then that's a recessive trait. So you would, both your parents had to have recessive alleles for freckles, and you got them both. So you won't be passing any freckles down to your kids, obviously, unless your spouse has, the freckle gene. Same thing with having a straight hairline, that's purely recessive. And having attached earlobes. So if you have earlobes that are attached to the side of your head rather than wiggle free, then that is purely recessive. And either A, both your parents have, attached earlobes and are purely recessive, or they carry the recessive gene for that. And you got both of them, one from mom, one from dad, and now you've got attached earlobes. So we can say that the dominant phenotypes, remember, dominant can either have, can be dominant, homozygous dominant, or they can be heterozygous dominant, right? Because it doesn't matter, we can't see that recessive if it's dominant versus recessive. So having freckles, either I could have, big A for freckles if both my parents had freckles, okay, or I would be big A, little a for freckles, and that's actually what I am because my dad doesn't have freckles, but my mom does. Okay, so I got the dominant from my mom, recessive from my dad, and I can actually see it in my kids. 2 out of 3 kids don't have freckles. So there you go. Now, recessive phenotypes are always going to have the homozygous recessive, right? Little a. Because if it's a recessive trait, you got to have 2 of the recessive alleles for it to show. Otherwise, it would be masked by something dominant. Okay, so this is kind of the fun part, at least for me, always with genetics, is to take a look at what's called family pedigrees. So you can take a look at the different traits as your family history has unfolded. So in humans, we can't necessarily do test crosses, okay, to analyze the genetics of some person. That would be kind of weird. But we can analyze, the genetics of people and kind of get an idea of what their genotype would be by taking a look at the family history of the traits. So we can construct what's called a family tree or a family pedigree, and then analyze the genotypes from what we see, from who has, that trait featured versus who doesn't. So we take a look at the pedigree on the right. Okay, this is for the trait of deaf versus hearing. Okay, so we started out with, our P1 or our grandparent generation of everybody is hearing. Okay, now Joshua and Abigail had a deaf son. Okay, so, but they also had, a hearing daughter as well, right? So, because they had one deaf son and one hearing daughter, and they're both hearing, that means that the deafness must be a recessive gene, a recessive allele of that gene. So we can assume that then the parents were both big D, little d, okay? Because both of them had to have that recessive in order for one of their sons to be deaf, okay? And of course, one of them had to be dominant in order for their daughter to be hearing. Okay, so we can get that kind of from there. Now, if Jonathan, if the deaf guy Jonathan is going to mate, and he mates with a hearing lady, okay, and we're not sure, whether she carries a recessive gene or not, right? It's kind of, playing the slot machines in Vegas. We're going to have to figure that out by looking at their offspring. So if she marries a deaf guy, it just depends on her genotype, and she's not sure whether she's going to have deaf children or not. So she had 1, 2, 3, 4, 5, 6. They had 7 children together, and 2 out of the 7 were deaf. Okay, so obviously she had to have the deaf gene. She had to be a carrier of that deaf gene. So she must have been big D, little d, okay, in order for any of her children to be deaf. If she was big D, then all of her children would be hearing, and we wouldn't see any deafness in that generation. Okay, so we can kind of go back and analyze different features of our family by looking at family pedigrees and kind of figuring out the genotype by looking at the different phenotypes and combinations from our family history. So, see, like we said before, that in order to have a recessive phenotype, okay, you always have to have a recessive genotype. You always have to be homozygous recessive in order to see that recessive phenotype. Dominant phenotype, on the other hand, is where you don't know. You are tricky because you could either be homozygous dominant, right, big D, or you could be heterozygous, okay, with a big D, little d. And so we won't know that until we look at your offspring, or perhaps even the offspring of your children, in order to figure that out. So some human disorders are controlled by single genes. It's just one gene that is going to control whether you have this or don't. So for example, albinism, okay, which is the lack of pigment, the people that have no pigment in their skin whatsoever, their hair or their eyes. So they are like purely white, they have white hair, and they're going to look like they have like this reddish color eyes. That's what we call albino, and that is going to be controlled by a single gene. It's recessive. So that means, the parents could be totally normal, skinned and pigmented, but if they both are carriers of a recessive albino gene, they could have a kid that is albino. Another one is cystic fibrosis, okay, or CF for short. It's where they have, it's recessive, excess mucus in the lungs, in the digestive tract, and stuff like that. It's not a fun disease at all. There's other ones, PKU. If you look on the back of soda cans, it'll say warning for PKUs. It contains phenylalanine, which is a amino acid that people with this genetic disorder cannot break down in their body, so they have accumulation of it. And it can cause mental retardation and weird skin pigment and stuff like that. So we call that PKU, or phenylketonuria. Okay, so again, it's recessive. Sickle cell disease, you probably heard of, is recessive. Tay-Sachs disease, which we talked about when we talked about the cells. That was the one where the lysosome disorder, right? The dysfunctional lysosomes of the cells is going to make a lipid accumulation in the brain. We get mental deficiencies, usually death in childhood and stuff like that. And there's also some dominant disorders as well. Okay, achondroplasia, which is dwarfism, is actually dominant. Okay, so if you are not dwarf, then you are little d. Okay, you've got the recessives. One type of Alzheimer's they have found was dominant controlled by a certain gene. Huntington's disease is another disease that is actually onset later in life. So unfortunately, people with Huntington's disease don't really know that they have it, know that they've passed it down until they've already had children. But basically, it's mental degradation in kind of the Middle Ages, usually starts in the late 40s and stuff like that. So these are all disorders that are controlled by single genes in your chromosomes. Most other disorders are what we call polygenetic, meaning, and a lot of other traits actually that we have in our bodies, eye color, hair color, and stuff like that, are all polygenetic. It depends on the combination of some of the genes. And you're going to see that different disorders are going to affect different groups unequally. For example, the cystic fibrosis, which is the excess mucus in the lungs and digestive tract. Your chances, if you're African American, your chances of getting it is 1 in 17,000, right? In Asian Americans, it's 1 in 90,000. However, if you're a European American, your chances are 1 in 1,800. So you can see that it affects different racial groups differently because of the probability. People, well, at least back in the day, it's not so much these days. We have a lot of biracial mixtures of having offsprings and stuff like that. But back in the day, it was usually kind of white people bred with white people, and African American people bred with African American people, and Asian people bred with other Asian people because they couldn't go across the world very easily. So that's why we see a different different disorders affecting different groups unequally. It'll be interesting to see now with more racially combined people, if these actually are kind of go away to the wayside. So we'll have to take a look in the next couple of generations to see how this is affected. Okay, so Mendel, super genius, of figuring out some of the genetic inheritance patterns and how basic genetics works. Okay, and however, if it were all that easy, it would just be super simple to kind of figure out, okay, who do I need to mate with in order for me to get a kid to look like this, right? However, it's not that easy. Mendel's two principles are only valid for sexually reproducing organisms when the characteristics are clear-cut, right? Flower is either white or purple. The seeds are either green or yellow. It's clear-cut when it's just two different variants. However, lots of our traits are more complex. We have more variants than that. For example, like I had mentioned before, we have different hair colors. We've got black, we've got brown, we've got blonde, we've got towhead, we've got red, we've got strawberry blonde. It's like we've got a whole bunch of different variants that we can deal with. So this is where we're going to start taking Mendel and kind of going beyond, okay, looking at what can happen when it's not just clear-cut dominant and recessive. Okay, so here's one thing. So we've talked about dominant versus recessive genes, but that doesn't happen all the time with certain characteristics. We can have what's called incomplete dominance, all right, where we have basically a variation between, like a phenotype that is between the dominant and recessive. So as an example, a simple example on the right here, we've got here a red flower mating with a white flower, and they make pink flowers. Look. Okay, because red and white make pink, so we get a variation between the dominant and recessive traits. Okay, so that's one example in flowers. A human example would be a condition called hypercholesterolemia, which is the accumulation of cholesterol, the LDL, in your blood. And we'll talk about that in a few minutes. Or now, we'll talk about that now, that's good too. So hypercholesterolemia in humans exists either in, normal, which is homozygous dominant, okay, big H, and basically what this means is that if you're normal, big H, then your cells have the ability to make receptors for this cholesterol, okay, this LDL cholesterol. And so your cells make a good amount of it so that this cholesterol can go from your blood into your cells, and your cells is able to break down that cholesterol and make, remember cholesterol is a lipid, so it can either use the cholesterol in making different hormones, or it can break it apart and use it for different, membrane, not proteins, but membrane lipids and stuff like that. Okay, so that is normal big H. Well, if you are heterozygous for hypercholesterolemia, okay, so you're big H, little h, you still make some LDL receptors, but not enough. Okay, so you might have higher cholesterol genetically because you are big H, little h. Okay, so this, this might be you if you've got kind of, high cholesterol, but it's not too high, it's not too low. You can, kind of control it with diet and stuff like that. However, there are some people who have this severe disease. They're homozygous recessive, little h, and their cells have the inability to make these receptors at all. So they have this accumulation of LDL cholesterol in their blood, which can lead to clogged arteries and heart attack and stuff like that. So a lot of times, you'll see in commercials about cholesterol is not all about what you eat and stuff like that, and that's true. It can be. There is a genetic component to cholesterol, and if your body is just genetic, because of genetics, is not making many of these LDL receptors, then you're going to have high cholesterol in your blood, no matter how healthy you eat. And then, of course, there's those normal, these people that are quote unquote normal, that can eat. It seems like they can eat whatever they want and they still have good cholesterol. So there's always genetic components we're finding to almost everything that we can see. All right, so we talked about incomplete dominance, where we get a blending, okay, of the dominant recessive phenotypes that we see. We can also have what's called codominance, okay, where both things are seen, okay, both of the the genes or the alleles are seen in the phenotype. And ABO blood typing in humans is codominant, okay. We can get 4 different blood types as a result from the combination of 3 alleles, okay, or 3 different forms of the blood type gene. Okay, so here are the 3 different alleles. We can have an A allele, okay, or AI, we call it. IA, I should say. There's a B, okay, which we call IB. Or there is O, okay, which is recessive, so we'll call that little I. Okay, so we can get people that are A-type blood because either they have 2 AI or IAIAs, or we have an IA with a recessive O. Okay, so that would be that would give a A-type blood. B-type blood is in a similar manner. Okay, you can have either big B, or big B with a little I. Okay, or the recessive. And you can have O-type blood. If you have O-type blood, then you must have both the recessive genes. And then we can also have AB blood, okay, which comes from a combination of codominance of both the A and the B alleles. Okay, so we can have that codominance as well. When we talk about genetics, we can also talk about what's called pleiotropy, which is the impact of a single gene on more than one characteristic. So basically what that means is that we can have a single gene have all kinds of effects on the body. So for example, sickle cell anemia is a disease that affects 1 in 500 African American children. 1 in 10 African American adults is heterozygous, so they're a carrier. So what sickle cell means is that basically if you have the sickle cell, it's recessive, so you have to have two recessive genes in order to be fully, sickle cell. You're going to make a deficient or an abnormal hemoglobin. Okay, so that your cells become, looks like, back in the olden days they had these kind of, they kind of used them as weapons too, but they were mostly like farm tools where it was like a sickle shape. It was like a knife that was kind of a C-shaped. Okay, and that's kind of how it got its name. The blood cells now become these little C-shaped kind of things instead of nice and round and big sphere shapes, or circles, I should say. Circles with a little dimple in it. Anyways, so pleiotropy just basically means that because you've got these sickle cells, they can't carry oxygen very nicely. You're going to have a whole bunch of effects from it. Okay, you can't break down, red blood cells very easily, anemia, paralysis, kidney failure, brain damage, like all kinds of things. So that's what we call pleiotropy. Kind of last but not least, polygenetic inheritance. Okay, so this basically means that a lot of traits that we have are polygenetic, or kind of the additive effect of two or more genes on a single trait. So basically it just means that two or more genes are controlling that single trait. Okay, so this, a couple of examples: height and skin color, eye color, hair color. Okay, these are all polygenetic. It's not just a single gene that's going to, affect the trait. And we can also have environmental effects on the phenotype as well, right? You go out in the sun, you're going to get darker skin. Okay, not because it's genetic, but because that's your body's response to being out in the sun. Your diet can also have an effect on your height and stuff like that. So we can have environmental effects as well. But like I said, a lot of our traits are polygenetic. In fact, I can actually see it in my own family. I'm blue-eyed. My husband's brown-eyed. Okay, we have two kids that are brown-eyed. Not really, that's not really surprising, right? Usually we think of, brown-eyed being dominant, blue-eyed being recessive. Then I have my third kid who has like these greenish blue gray eyes. Okay, so not clearly blue, not clearly brown, not really in between. So there's got to be more than one gene that is affecting eye color, and that's exactly what we have found. It is actually just more than one gene affecting eye color, and the same with hair color as well. So it's kind of cool when we kind of take all that and blend it together, and then we get totally different traits. It's kind of cool. Okay, so kind of a unifying theme that we have touched on throughout learning about meiosis and patterns of inheritance is this chromosomal basis of inheritance. That chromosomes, the chromosome theory of inheritance states that genes are located on chromosomes, right? And that the behavior of the chromosomes during meiosis, meaning, how they line up and which daughter cell they go to and stuff like that. And then also in fertilization, the random chance and probabilities of coming together with certain, eggs and sperm is going to account for inheritance patterns. So we have this, whole big gamut of how we can get different inheritance patterns with, single, basically single genes and how they go through meiosis and fertilization. Chromosomes undergo segregation and independent assortment during meiosis, and so account for Gregor Mendels Principles. Okay, so we can see that occurring when we take a look at different genetic traits. We can see that we get the segregation of different chromosomes into different things, and they kind of segregate independently. So it's not that we always have, if the gene for eye color is on, just say we'll assume that eye color is one gene. Okay, so the gene for eye color is on one chromosome, and the gene for freckles is on the other and on another chromosome is not necessarily that they go together all the time, right? Sometimes we can get the freckle gene going in one cell and the eye color gene going in another cell. So that's our independent assortment. Okay, and so again, Mendel could see that, and we could also see that. This slide here is basically a nice kind of meshing between Mendel's principles and knowing stuff about chromosomes, genes, and DNA. Okay, so we can apply what Mendel saw at each step, and also, put in our knowledge of DNA, chromosomes, and how they behave as well, and get the same results. Okay, so this is basically what this slide is about. And in the next couple of slides, we'll kind of put this into words that you can easily write down. But basically you got this P generation, right? If we cross a yellow round seed plant with a green wrinkled seed plant, okay, we can get meiosis, fertilization, blah blah blah. We get this F1 generation that are all round and yellow seeds, okay, because they all have the same genotype. If we start out with two purebreds, we are going to get a first generation that all have the same genotype, which is basically a combination, big and little, of each parent. Okay, so if we take a look at the particulars of meiosis of the F1 generation, we can see the principles of segregation, of the chromosomes segregating into different daughter cells, okay, as well as independent assortment, meaning that there's a bunch of different ways in which the chromosomes can sort. Either, all of the, for example, all the red ones can go to the left and all the blue ones can go to the right, or we get a combination of red and blue to the left and red and blue to the right. It just depends. They're kind of independent. It just depends on how they come together and during meiosis. Okay, so we get a whole. We can see that we can get a whole bunch of combinations, and that's exactly what Mendel saw in his F2 generation. He saw a whole bunch of combinations of phenotypes in those F2 plants, right? So, and it actually, interestingly, always occurred in this ratio, 9, 3, 3, 1, okay, where 9 of the offspring are round and yellow, 3 are round and green, 3 are yellow and wrinkled, and 1 is purely recessive, green and wrinkled. So this is again a meshing of our understanding of DNA and chromosomes as well as Mendel's principles, and you can see that they can clearly go together. So this slide is basically going to pick out the main points of what I just said in the last slide so that you don't have to dictate everything I said in the last slide. So here we've got in our P generation, we've got our homologous chromosomes that can separate during meiosis, get recombined to make an F1 generation. Right? And then these F1 generations, because of independent assortment and the principle of segregation, can make a whole series of different kinds of gametes, right? That can come together in fertilization randomly to make an F2 generation with a bunch of different, a lot more different combinations that we had saw previously. In fact, these combinations again come together in a nice ratio, 9, 3, 3, 1. 9 being homozygous dominant, the 1 being homozygous recessive, and the 3 a combination of the 2 in between. So the field of genetics is hugely complicated, and here's another wrench to throw in to our genetic study is gene linkage. Okay, so genes located close together on a chromosome are not actually going to follow Mendel's principles of independent assortment. Okay, so what that means is that in Mendel's observations and with the pea plants, the purple flower was on one, the gene for flower color was on one chromosome, and the gene for seed color was on another chromosome, okay? So that's how we could get purple flowers, and so when they separate, that's how we could get, purple flowers in one with this kind of seed versus white flowers in the other with the other kind of seed, okay? We get different combinations. That was, if they're on separate chromosomes. But there are some genes that are located close to each other on the same chromosome, okay, like those. So remember, the individual chromosomes don't break apart into little pieces as they go to the side, okay, as they split apart. They're actually going to go together. So chromosomes that are on, or I'm sorry, genes for different traits that are on the same chromosome will be inherited together, okay? So they're not going to follow the law of independent assortment where we can get a variety of different seed color and seed shape, okay? So, if they are linked on the same chromosome, then that means that every time you saw a certain color, you would see a certain shape because they are linked to the same chromosome. So that's another kind of wrench that makes genetics more complicated. We've already talked about this one before when we talked about meiosis, but remember the concept of crossing over, okay? During meiosis, when those tetrads of the homologous chromosomes come together in metaphase 1 of meiosis, we can get the crossing over of, in the breaking apart and recombining of different genes on the sister chromosomes. Okay, so that can produce, haploid daughter cells with a completely different combination of alleles. It could be mostly mom's genes on that chromosome, but it'll have, a dad little counterpart because of crossing over. So that's another complicated thing as well. The last concept we're going to talk about in genetics is the sex chromosomes and sex-linked genes. So remember, the sex chromosomes are going to determine whether you are male or female. If you have an XX combination, you are female. If you have an XY combination, you're male. So this is a homologous set of chromosomes, but on these chromosomes are other genes that control other traits as well. So we're going to have what are called sex-linked genes. Any gene located on the X chromosome is called a sex-linked gene. Now, X chromosomes are much larger than Y chromosomes and contain, many more genes than the Y chromosomes. So sometimes, like we can see when studying Drosophila or fruit flies, that you're going to have an absence of certain characteristics in males just because the Y chromosome doesn't carry that gene for that. So it'll be, kind of dominantly whatever the mom had, the son is going to have. Okay, so most sex-linked genes are usually actually unrelated to sex determination and are found on the X chromosome. So whatever, your mom had on her X chromosome, the males are going to portray because the Y chromosome is a lot smaller and probably doesn't have anything to counter, any other allele to counter whatever's on the X. So it's kind of like whatever mom's got's gonna just be dominant because the Y doesn't have that. So in humans, some of these sex-linked genes include, unfortunately, some disorders, like red-green color blindness. So here's a picture. If you're red-green colorblind, you would not be able to see the number that is, inside of this circle. It's a different color than the other circles. So that's found on the X chromosome. So if your mom is a carrier for red-green color blindness, the males are going to show it, and the females won't. They'll just be carriers. Hemophilia is a disease of the blood, unable to clot the blood efficiently, so you can basically bleed to death. And again, that is a sex-linked disorder found on the X chromosome. So it's highly seen in males where the mom is the carrier again. So females usually don't show these characteristics unless they have like the unfortunate combination of a mom being a carrier and dad actually having the disease, and then a female can have it, but it's very rare, very rare. And then the last one, Duchenne muscular dystrophy, a disease where a complication or disorder, miscommunication between nerve cells and the muscle cells that they can't kind of relate or communicate back and forth, and so we get muscles not forming correctly and not functioning with the brain correctly as well. So again, that one is a sex-linked disorder on that X chromosome. All right, so that is going to wrap up our discussion of genetics, meiosis, mitosis, and patterns of inheritance, and how we get different gene traits and phenotypes and genotypes. So hope you had some fun.